Well, hello everybody. 17.2. Here we go. So 17.2 is all about the transcription and about how transcription is a DNA-directed synthesis of RNA. Okay, so remember, let me recap 17.1. As we began talking about protein synthesis, the process of transcribing DNA to mRNA and translating mRNA to protein. Okay, today we're going to focus on the transcription portion of DNA to mRNA to be sent out into the cytoplasm to become a protein via translation. Okay, so we're going to focus on this part, and it happens within the nucleus. All right, here we go. So I thought a long time about what's the best way to illustrate all the pictures and how they flow together, and it's actually through an 8-step process. Okay, so follow me through here. You can pause it, you can write it down, do whatever you need to do, or you can just listen. So transcription is run by an enzyme called RNA polymerase. RNA polymerase is very similar to DNA polymerase in that, yes, it can add all the base pairs, etc. But RNA polymerase also has different enzymatic activities, such as helicase and even topoisomerase. So we don't have those enzymes per se, but we have this main enzyme called RNA polymerase that does everything. And RNA polymerase then uses one strand of DNA, because remember, RNA is single-stranded, versus DNA is double-stranded. So it's going to pick one of the strands to use as a template to form RNA. In this picture, we see the RNA polymerase coming in and doing that job. It's going to unwind. It's going to go ahead and start doing its job here. Here's the start point. We see how, as you progress through this, there's a binding, there's an initiation, there's an elongation of creating the mRNA, and then finally there's a termination. If we zoom in here, for the elongation, we see that very similar to the DNA polymerase, it's going to add the complementary pair. So we're going to use this DNA strand, this bottom one, as the template. So where there's a T, we're going to put an A. Where there's an A, we're going to put a U. Where there's a G, they're going to put a C. So this is very similar to the last video that we talked about transcription. Okay, adding finding the pair to the DNA, the RNA pair. Okay. As you kind of saw in that last picture, RNA polymerase, with the help of transcription factors, find the promoter of the gene by looking for a particular series of bases that pretty much tell the enzyme, hey, start here. Okay, so this is actually very similar to the primer that we saw in DNA replication. But now this is for transcription. So there's a promoter sequence. Okay, and then in addition, as we just talked about, RNA then begins reading the DNA and attaches nucleotides using U and not T. Okay, so let's take a look at what that looks like. So this is what I was referring to in class about the Tata box. The Tata box is a sequence of bases. As you can see here, TA, TA, AAA. And pretty much this is a signal for the transcription factors to combine, and then the RNA polymerase then to know where to start transcribing. And at that point, it's just going to keep going, and then it's going to create a single-stranded mRNA from it. As we move into 17.3, we talk about how it's not just transcription happening, but eukaryotic cells will modify RNA after that transcription happens. So let's see how that looks like. So after we transcribe that double DNA strand to a single mRNA strand, 5, during RNA processing, it's going to enter this process called RNA processing. Both ends of the primary transcript are altered. We're going to add on a 5' cap and a 3' poly A tail. So you got a cap and a tail to the mRNA strand. And this is to help the mRNA come out of the nucleus, the export of the mRNA. And it's also to help the mRNA be protected from degradation of like enzymes around the area. Okay, so here we see the 5' poly A tail, the 5' cap. We see this region that we're not going to have any coding genes in. Then this is our coding segment, the exons. And then, like we'll talk about what that means. And then we have this polyadenylation signal. So this is, once again, not going to be coded for, the UTR. These are the untranslated regions. And then we've got the poly A tail. So in this picture right here, the main thing you need to know is that this is the good stuff right here, exons. That's it. Everything helps together to push this out of the nucleus. Okay. Then, after RNA processing, before the RNA leaves the nucleus, it's going to go through one more process called RNA splicing. And RNA splicing, in order for RNA sequence to become functional, non-coding sequences such as introns are cut out of the sequence, and the remaining portions, exons, are put together. So let me show that. Let me show you how that looks like. So once again, introns are pretty much junk DNA. It's just a bunch of base pairs, a bunch of letters that really don't code for anything. Exons is the good stuff, and that's the express sequences which contains our genes, and that's what we want to keep. So introns are going to be out. We're going to try to cut them out, and then the exons, we're going to leave them in. Does that remind you of something? Introns out, exons in. How have I always memorized this? This is a little weird. Okay. Introns out, in and out. All right, here we go. So hear me out here. So here's how I've always memorized what goes out and what stays in. Introns being the non-coding sequences that don't have the genes, we want them out. So I've always memorized that by in and out. Introns out. Versus the exons, we keep in. And we put them together, and that's our gene that gets pushed out into the cytoplasm to a ribosome to create a protein. But all these introns are non-coding sequences. It even says in your textbook, let me see here. The average length of a transcription unit amongst human DNA is about 27,000 base pairs. 27,000. However, it only takes 1,000. That's like 1 in 27th time. It only takes 1,200 nucleotides in RNA to code for the protein. So we don't need 25,800 nucleotides. So that's why we got to splice them out. And how do we do that? Okay, so let's take a look here. So once again, exons stay in, introns are out. We're going to push out these introns, keep the exons together, put them together. How do we do that? It's through spliceosomes, and it's through snRNPs. Okay, so let me see here. Researchers, so this is from the textbook. Researchers have learned that the signal for RNA splicing is a short nucleotide sequence at the end of an intron. So what they're saying is at the ends of these introns, there's a little sequence of bases, and that tells these snRNPs, which stand for the small nuclear ribonucleoproteins, SNRPs, that's what they call it, to splice them. So there's these little base sequences here that tell these snRNPs to come in and splice them out, to cut. So now we can remove the intron and put together the exons. And the exons are important for us because they stand for the coding sequences, and they have all of our genes that we want to turn into proteins in translation. Hopefully this video helped. Once again, I don't mean to waste your time, but I just hope that these videos are helping and that I can help you understand these complex processes a little bit better. Have a good one. Positive attitude. Bye.